So good morning. This is how I see UNH. So forget all the snow that we've been having. So thank you for this opportunity for me to tell you about contributions of the ovary to fertility in dairy cows. And what I'm going to talk about, this presentation outline, is going to be describing for you the two important structures found within the ovary. And in particular, I'm going to be telling you about the vasculature, that is the blood vessel system of the ovary. And specifically, to tell you a little bit about the factors that are responsible for forming new blood vessels, a process that's known as angiogenesis. And then I'll review for you some of our previous and current work on identifying some of these factors that are responsible for forming new blood vessels in the cow corpus luteum as well as the cow follicle. And then to conclude, to tell you about how information from basic research of this type can be applied. Now, most of you, if not all of you in the audience, knows that reproductive performance in dairy cows rely on a number of factors, of which sire conception rate, nutrition, as well as estrus detection are really important. The focus of my talk actually is on the maternal side, that is maternal fertility. It is within the ovary that an egg is grown and then becomes competent. But that's not enough. The egg is, after it's released, it's fertilized in the oviduct. And further, if that is successful, then the fetus has to grow, depending on the gestation rate of whatever species we are talking about in the cow, about 9 months or a little more. They have to grow to maturity within the uterus. And so the optimal environment that's needed is required for that to actually occur. In our minds, one link of these three events, so to speak, is actually the vascular tree, or the blood vessel system that's found in the reproductive tract. Here is this very beautiful picture of the extensive vasculature of the blood vessel system in the cow uterus, as well as the vascular system that's associated with the ovary. And having such an extensive vascular system not only provides a means by which nutrition can be, nutrients can be delivered to the cells in the uterus, as well as the cells within the ovary, but that their products can also travel to different parts of the body. Now, the ovary is kind of hidden back here a little bit, and so I brought up another picture of the cow ovary, which shows a very prominent structure called the corpus luteum. And there's absolutely no doubt that there's also an extensive vascular system that is present in the corpus luteum. So speaking of the ovary, it is a dynamic endocrine gland, and what it does is that you find the two most important functional structures present, the follicle and the corpus luteum. So when you look at the ovary at any time, there are follicles of different sizes, and during a certain period of time, one follicle is lucky enough to be selected. And that selected follicle then becomes the pre-ovulatory follicle within which it's going to ovulate the egg. And so when the pre-ovulatory follicle grows and matures, it actually changes the way it looks. And so not only does it increase in size, but it's filled with fluid. And within this fluid-filled structure is where the egg is actually housed. So it protects it, and at the same time it nurtures it, because this follicular fluid is filled with goodies that the egg will mature and develop properly. When the hormone signals are appropriate or correct, then the egg is then released. So here are two pictures of cow ovaries. Here's one, and here are the follicles, the two large follicles. They are translucent structures because by and large they're fluid-filled structures. I want you to notice the reddish tinge on this, and that indicates that there is a very good vascular system associated with the follicle. Here in this other ovary, not only do you see a follicle, but then you see the other structure that I mentioned called the corpus luteum. And the corpus corpus luteum is one of the most angiogenic or tissues in the body that has a lot of blood vessels. So the corpus luteum actually comes from the follicle. So when the follicle develops and then the egg is released and then migrates to the oviduct for fertilization, this structure, the remaining parts of the follicle, kind of collapses on itself. It doesn't die, however, but instead what it does is do this amazing change, this amazing transformation to become a new structure on the ovary called the corpus luteum. And it is the corpus luteum that produces progesterone, which is the hormone of pregnancy. And so in a study done by Fields and Fields, what they did was to remove ovaries at different days of the cow's reproductive cycle, and then isolated the corpus luteum from each of these ovaries, and that's indicated here, and then they did a cross-section of it. So this would be indicative of young, developing corpora lutea. This would be indicative of what we call middle-aged corpora lutea, and these are old and then, if not dying, corpora lutea. So you could easily see how it increases in size very quickly, and then it maintains itself, and then when it dies off, it actually decreases in size. What I want you to note particularly is that how red this early developing and middle-aged corpora lutea are. And that has to do with the very extensive blood vessels that are actually present in the corpus luteum. You probably don't know that having new blood vessels be formed is very rare. It's extremely rare in normal tissues by normal cells. It is usually associated with actually diseases such as tumors. And it's from tumors that we learn about how blood vessels are actually regulated. So what the tumor does is to send out these signals, and these are growth factor signals. There's a ton of them. And it tells the parent vessel to say, hey, it's time to sprout new ones. And then the sprouting involves obviously a lot of things, but I'm focusing on these enzymes called matrix metalloproteinases, or MMPs. What they do is they need to clear a path between the parent vessel and the tumor. And so clearing that path then allows these sprouts to grow toward it, and then it will infiltrate the tumor, and then what's going to happen is that it's going to feed that tumor, because with its growing in size, that it's going to need a lot of nutrients. How can this apply to what we do in the cow reproductive system, in the cow ovary? So in the cow follicle, there are two cell types to remember, or to know, to be familiar with: the granulosa cells and then the theca cells. And you notice that it's the theca cells that actually have a blood vessel system. At this time, there's no blood vessel system in the granulosa cell. As it matures, and then when it's picked to ovulate, that is, the egg that's going to ovulate, what's going to happen is that this is breached here, and upon that breaching, something starts. And what that something starts is that you're going to now have new blood vessels sprouting from these parent ones, and then if you recall, this whole thing collapses on itself, and then it becomes the corpus luteum, this very vascular tissue. So we took the idea from tumors, and then now you see how I replaced it with luteal cells, or cells of the corpus luteum. And so we asked this question: are these enzymes, what I call the remodelers of our system, are they there to clear space for new blood vessel to be formed, and do luteal cells actually produce them? So the cow is a great model to study this, because not only can we get enough tissue, but we can actually look at development. We can get different ages of corpora lutea and see at what age are these enzymes made. And so this was a study that started us off. It was a while ago. And so what we ended up doing was to collect corpora lutea that were 2 days old, 4 days old, therefore collecting them every other day until they were 16 days old. And so this particular test that we use is pretty complicated, and to simplify it for you, with these red arrows, I've indicated these clear bands. Every time that you see a clear band, it means that the enzyme is present. Okay, so there's a significant production of this particular flavor of enzyme. However, there are many other flavors of this enzyme. So in the upper band, you might not see it very well. There are much thinner clear bands here, but what excited us really the most was the bright band that's at the very early stages of corpus luteum development. Remember, that's when it's very highly angiogenic, a ton of blood vessels are formed, and so it needs help. So there are multiple enzymes that are present. So what we ended up doing was we actually cloned the cow version of this particular enzyme, and we then went on to find what cells within the corpus luteum actually make this enzyme. Forget A, B, and C. Look at D, E, and F. And anytime you see the brownish red coloring, it indicates that that enzyme is present. The black arrows show pointing to this blood vessel and then to these blood vessels here. It shows that those endothelial cells that line the blood vessel, they can make this enzyme. So we went, phew, they can do this. But what was very interesting to us is that the cells that are pointed to by these white arrows, what they show is that the cells in the corpus luteum that make progesterone, they can also make this enzyme. And so you have multiple cell types within the corpus luteum that can make this enzyme. So it clears the way for new blood vessels to form. So we asked the question, do these luteal cells actually, what kind of signals do they send out? At the time, others have found all these different types of growth factors that were that reported that the luteal cells can make. However, with help from the New Hampshire Agricultural Experiment Station, we were interested in looking at this particular protein, abbreviated as CYR61. It is found in tumor cells, and it does a lot. But here, you see what it does? Angiogenesis, the growth of new blood vessels. And so we showed, therefore, for the first time, that this particular angiogenic inducer, CYR61, was actually produced by the cow corpus luteum. In this particular test, anytime that you see a dark band here in samples, it indicates its presence, and then the darker the band, the more it is produced. And so everything is kind of falling in place now, because here in the 4-day-old corpus luteum, you have a lot of this angiogenic inducer being produced. So all the players are present in the cow corpus luteum. So at the time, we were reasoning that you've seen this picture before of the blood vessel system in the corpus luteum. So what we reasoned was this extensive vasculature actually was born in the follicle. So remember how this parent vessel invades the inner compartment of the follicle, and then the whole thing then collapses on itself, and then it develops into the solid gland. So it was actually born there, and so we asked, therefore, the same questions that we asked in the corpus luteum in the follicle. So see how I changed that now? So again, we wanted to say, are these enzymes there? Are these demolitioners of tissue in follicle cells? And so let me explain the bottom part to you a little bit here. So when a follicle is selected to ovulate, it takes anywhere from 60 to 72 hours for it to develop and then ovulate. And so what we ended up doing was to actually look at what we call 0-hour, a 24-hour-old preovulatory follicle, and a 48-hour-old preovulatory follicle, and then looking at the two different cell types that populate the follicle. And clearly, you could see that the vascularized layer, that is the theca layer, produces in very high quantities this particular enzyme. We also wanted to know what are some of these growth factors that what are some of these messages that are being sent out by follicle cells. So we looked through a lot of them, and this is just a representation. Again, it's in the same presented to you in the same way: 0, 24-hour, 48-hour-old follicle, and then granulosa and theca cells. And so the take-home message here is that these cell types have the ability to make, at least in this particular case, a growth factor called fibroblast growth factor. Although there, at this time, we don't see any differences between the different ages of the follicle. So you might wonder, what about the CYR61, the one that we were so excited about from the cow corpus luteum? Here in the granulosa and the theca cell of the cow follicle, we also show for the first time that this angiogenic inducer is highly expressed in the theca, but is also expressed in the granulosa. So what do we make of all of the data that I've just presented to you? You could, so this is a summary of our work as well as others in the follicle that show the granulosa cells, the theca cells, remember that's the layer with the vasculature, and the messengers and other enzymes that are produced by these cell types. And now what we could do is actually add, based on our work, that this angiogenic inducer is produced both in the granulosa and the theca cells. Now, what we don't know yet is what's actually driving the production of the CYR61, as well as the other angiogenic factors in the cow follicle and corpus luteum. So in other words, how is, what does LH and FSH, LH standing for luteinizing hormone, FSH standing for follicle stimulating hormone, these two hormones are the key regulators of follicle function. That is that they will regulate steroid production by these follicle cells. What we don't know is what role they play in regulating the CYR61 and other growth factors and how they impact the development of blood vessels. So to conclude, what does this all mean? Here's all this basic information. How can we actually apply it? And so, in my dream world, I'm hoping that the CYR61 plays an extremely important role in generating this blood vessel system and in regulating the development of new blood vessels. And so, in a way, I actually want to develop an angiogenic signature. And that angiogenic signature can then be used to screen for maternal fertility in terms of, let's say, distinguishing between cows of high fertility versus cows of low fertility. So wouldn't it be nice to take a blood sample and be able to say, yes, combined with maternal fertility and, of course, the other factors that I've mentioned, that is going to enhance reproductive performance in dairy cows. So a study like this requires a lot of people currently in my lab, as well as others, former lab members, as well as collaborators, my dear friend David Townsend from UNH, as well as my two collaborators from the Children's Hospital. They actually are a cancer lab, and they see the cow model as a great way to actually learn about cancer growth and how actually to stop cancer. And of course, the study cannot be done without the cows, as well as the wonderful staff at the Fairchild Dairy Teaching and Research Center, and of course, funding from the New Hampshire Agricultural Experiment Station through multi-state projects in 1027, 1227. Thank you.
We are running a little behind, and so there's time for a quick question, but I'm sure Paul would be happy to take other questions afterwards.
Dr. Erickson.
Any connection with your work on the follicular waves with the CYR61?
We haven't looked at that, and we actually, through our multi-state project, one of the studies that we did was actually show, so let me step back. when follicles develop in animals like a cow, they actually develop in waves. That is, that they have a cohort, and then within that cohort, one's actually selected, and then it grows. But since the hormone environment is not correct, then that follicle kind of dies off, and then a new cohort develops. And so so cows can exhibit either 2 waves of such growth, or 3 waves of such growth, or actually 4 waves of such growth. And so through our multi-state project, we actually found that cows that have 3 waves of growth actually has much higher fertility than 2 waves of growth. But we haven't looked in terms of how it relates to the CYR61. But I think this is all part of the maternal fertility that I talked about, and how, as I said, in my dream world, I love to take a blood sample, and I want to tell you that that cow has the angiogenic signature that will be indicating high fertility.